Please welcome to the stage meeting co-chair Alexandra Warden.
Hi everyone, welcome. Thanks for being here late in the evening after a long day of science, hopefully very exciting. I just want to remind you all that you can use the translation service to hear Canadian French using Wordly in this session. I also want to remind you all, hopefully there will be a slide in just a moment, to join us tonight for the Rainbow Reception, which is to celebrate a network with our LGBTQ+ community. I also want to remind you, for those of you brave enough to run up a very large hill tomorrow, although I'm told the scenery will be great, that if you'd like to do that fun run and travel with a group to it, please be outside the southwest doors, that's near all the colorful glass, at 6:45 a.m. And transit tickets will be provided if you need them. And so lastly, I'd like to introduce Barbara Barta, who will be presenting the SIL's Kilham Award to the recipient.
Hello, good evening, and welcome to the Kilham Memorial Award lecture today. First, I'm going to say a few words about the award and then introduce the excellent researcher who won this honor this year. So the Kilham Memorial Award was established to honor the memory of Peter Kilham by promoting and stimulating research in the fields that most inspired him and to which he devoted his life to. These include African limnology, the ecology and physiology of planktonic diatoms, comparative ecology of lakes and oceans, amongst many other topics. He was an extraordinary scientist whose work really shaped modern limnology. Born in England and raised partly in Uganda, he developed an early fascination with aquatic systems and the natural world. He completed his PhD in zoology, focusing on the chemistry and biology of tropical African lakes, and throughout his career, he worked at many renowned institutions. Peter Kilham was widely admired not only for his scientific insight, but also for the generosity with which he shared ideas and collaborated with colleagues all around the world. The Kilham Award, therefore, became one of the major honors of our society. It recognizes scientists whose research embodies the same spirit of curiosity, synthesis, and broad ecological vision. This year, the committee received 11 outstanding nominations, reflecting the remarkable breadth and quality of research within our community. On behalf of SIL, I would like to thank the members of the Kilham Award Committee for their time and the thoughtful evaluation they devoted to this process. It must have been a hard task to choose amongst the exceptional pool of researchers. So I would like to thank Sarian Kosten, Stuart Bunn, Jasmine Saros, Björn Wissel, and Maria de Los Angeles González Sagrario for their work. And now it is my pleasure to introduce the 2026 recipient of the Peter Kilham Memorial Award, Dr. Silvia Bonilla. Dr. Bonilla is an associate professor at the Limnology Division, Faculty of Sciences at the Universidad de la República Uruguay. She is internationally recognized for her contributions to phytoplankton ecology, particularly the ecology and ecophysiology of cyanobacteria. Her research spans a broad range of ecological scales, from physiological mechanisms to continental scale patterns, from pigment analysis to empirical predictive models, and from shallow freshwater lakes to brackish coastal lagoons. Her work has advanced our understanding of phytoplankton community dynamics, particularly in relation to anthropogenic stressors, for example, nutrient enrichment, and to regional climatic differences, temperature, and hydrology. She has also contributed significantly to international collaborations addressing cyanobacterial blooms and water quality all across the Americas. And actually, she finds these international collaborations as one of the most enjoyable parts of doing research. She is the first woman to receive a PhD in limnology in Uruguay, and since then she has pioneered limnological studies in her country. Currently, she is the director of the Limnology section of the Faculty of Sciences at her university, and as the current president of the Uruguayan Society of Limnology, she is contributing to evidence-based decisions and promoting the diffusion of limnological knowledge into other sectors of society. She makes frequent media appearances to discuss issues related to aquatic ecosystems and algal blooms, and she works tirelessly to raise public awareness of the importance of protecting freshwater resources. At SIA, she is a foundational member of the working group Cyanat, controlling cyanobacterial blooms through nutrient load reduction. And beyond her scientific accomplishments, she is recognized as an inspiring mentor, collaborator, and leader within our community. Her work really exemplifies the integrative and collaborative spirit that characterized Peter Kilham's own scientific life. So please join me in congratulating Dr. Silvia Bonisha, recipient of the 2026 Peter Kilham Memorial Award.
Hello. Thank you. It's for me a great, great honor to be here today and a great pleasure to be back in Quebec. I would like to thank the colleagues that nominate me for this award. This award is in name of Peter Kilham, as we hear in the introduction. He was a remarkable scientist that made a significant contribution in our understanding of tropical lakes. And still today, there is a lot of work that needs to be done with warm tropical lakes. I come from Uruguay. Uruguay is a small country in the southern corner of South America. And I also come from this reality, where we can see in this plot is that all countries in South America and the Caribbean are at the end of the list of investment in research in relation with the GDP percentage. So research in this region is a challenge. That's why I would like to share symbolically this award with my colleagues in South America that work in harsh conditions with so much passion and stamina. I am a very lucky person, and I have the pleasure to work for so many years with great mentors, inspiring colleagues, and motivated students that make my work really joyful. So thank you to all. Today I'm going to talk about cyanobacteria, and I don't need to convince you that cyanobacteria are fascinating organisms. I will say maybe one of the most fascinating organisms to work with. They have a size range that cover more than 3 orders of magnitudes that makes them available to success in different trophic levels and different environments. They also, among the photolithotroph, are the group that has the highest diversity in pigments that allow them to respond to different light environments. And this is an aspect that needs much more research in limnology. But of course, cyanobacteria had the bad side. The dark side of the cyanobacteria are the cyanobacterial blooms, that of course they have a public health consequences and ecosystem, also at the ecosystem functioning. So today in my talk, I will have 3 parts. In the first part, I will talk about cyanobacteria in general, as the group, and the abiotic factors as are related with blooms. Then in the second part, I will talk about different cyanobacteria, and I will give examples of some of them. And at the end, I just left very few slides because of time constrictions, but I would like to show you a little bit about challenges of monitoring cyanobacteria in South America. So I started with the first part. I want to share with you today a work that was very nice, and we did with colleagues from 11 institutions in South America. We built up a database for South American countries I think it's still the biggest and the largest database we have for phytoplankton in South America. And then we complemented that information with data from the United States and also from Canada. And we studied the distribution of cyanobacteria. And in this first map, here is the biovolume as an indicator of biomass, and here we have the latitudes, and we can see that despite the 5 climates that we cover, there is no clear trend in the cyanobacteria biomass distribution with the latitude, except for the very extreme polar regions. In this plot, at the left, we have the same information but grouped by climate, and here we can see that polar and equatorial tropical lakes, they have lower biovolume than the other climates. But I would like you to pay attention in this plot, where here we have the dominance, the percentage of cyanobacteria in total biovolume, and here we see that warm climates, they have a higher proportion of cyanobacteria. So in this plot to the right, here we have the biovolume of cyanobacteria and the proportion of cyanobacteria in total biovolume. And what we noticed that is quite remarkable is that cyanobacteria can be dominant in tropical lakes, but a very low biomass. And that is a natural condition for these kind of lakes, usually are related with low trophic levels, even in oligotrophic lakes, where picocyanobacteria can be dominant. So the dominance of cyanobacteria in these lakes, in tropical lakes, does not mean high biomass or a problem with cyanobacteria. Then we start looking at factors behind this biomass distribution, and here I show you all these 3. We could find, this is just the description of the distribution of the data. We have a positive trend, a positive relationship with phosphorus and nitrogen, and we didn't find any clear trend with temperature. We performed many different models, and I will show you this case at the end, where we have here, we split the lakes in 3 categories. In red are shallow lakes, in green are middle depth, and in blue are deep lakes. And we found that phosphorus was the major predictor of cyanobacterial biomass, but nitrogen became significant, especially for shallow lakes. However, temperature was never a significant factor in any of the models we tried. So a big message we have here is if we have nutrients, we can have cyanobacteria and high biomasses of cyanobacteria, despite the climate and despite the latitudinal gradient. And the second message here is that it depends also on the lake type. In this case, we have these categories of depth that can make a big influence in the relative importance or influence of one of the nutrients in the distribution of cyanobacteria. And these are the kind of questions that we are working and discussing in our SIL working group for the last 2 years. This group is led by Ingrid Korus, and it's called Cyanut. And for me, it is a great pleasure. I've been learning so much working with so great scientists in this group. And so we hope that this year we'll have nice review papers coming out. So probably you will see them soon. So the kind of work we are doing now is compiling information that is in the literature, evidence, to play around with this idea. In here, just as an example, we have total phosphorus and cyanobacteria, and we expect to have a kind of relationship like this. So what we started looking was at what are the conditions that can change the biomass yield in this axis. And here I just mentioned a few. We have many more in this review paper that we are working on it. But I would like you to pay special attention to the ones related with tropical lakes. And here we have dry seasons and rainy season. And the 2 seasons can have opposite trends and opposite influences in cyanobacterial biomass. And the other set of factors are related with the conditions shifting where the relation biomass and phosphorus curve level off. And here we have other kind of factors. And again, I would like you to pay special attention in the case of tropical ecosystems where omnivorous fish, which are very, very common there, obviously, they can make the situation much more complex and less predictable in respect with cyanobacteria. So before finishing this section, talking about cyanobacteria in general and the factors, I will invite you to come with me to another scale and look at the organisms and nutrients in the relationship with dissolving organic forms, which is the nutrients that the organisms will really uptake. So we have to think about the dissolving organic nitrogen and the soluble reactive phosphorus, and in this step is really about the nutrients and the organisms. There are two facts that we need to accept here, and one is that nutrient uptake it's a flexible process. There is not only one way or one amount that the organisms will incorporate. Is flexible. And the second one is that the uptake can occur until a minimum, a limit that we call it threshold, below what is not energetically able to keep going that uptake. So with these two assumptions, I would like to show you the results of some experiments that we performed led by my colleague Luis Obriot in Uruguay. Here we have, we have a problem. Okay. So we perform experiments, uptake experiments with natural phytoplankton from an eutrophic lake dominated by Plantotrix agardi bloom, Plantotrix agardi and Rhaphidiopsis mediterranea at that case. So I would like you to pay attention to the plot to the left, where we see what was in the lake in different moments. We have 37 experiments here. So we have the ratio, the nitrogen and phosphorus ratio, and there in the other axis we have just the concentration of phosphorus. So we can see that phosphorus becomes more and more limiting as we go to the right. And then then I would like you to pay attention to the second plot, to the one in green, and what we have here is what the organisms did. So what we have here is the threshold up to when the organisms incorporate phosphate. So those are the values where they stop incorporating phosphate. And here we have two important messages. First, they can incorporate phosphate very, very low, near 0.1 micrograms per liter. Actually, the minimum we got was 0.17 micrograms per liter. That's one message that's very important. And the second one is that nitrogen is a mediator when phosphorus is stressful. So if nitrogen is there, they can keep incorporating phosphorus to very, very low levels. And that means that the importance of the interaction between the two nutrients can explain the resilience and the resistance of blooms when phosphorus is becoming very, very limiting in the dissolved form. So finishing this section, we are going to see about the species, or who are behind these general ideas of cyanobacteria as a group. And now we have problems with the pointer. Okay. Yeah, so when we talk about blooms, and that is a discussion that I would like to have in another plenary, maybe, that what is a bloom? I've seen different ideas of blooms in this conference, very interesting, and with different levels of biomass and different conceptions of blooms. So, but we can talk about that another day. We can organize blooms in three large groups in relation with the energy requirements and the stability of the water column. And we have the accumulate ones, the ones that can be accumulated at the surface or accumulated at depth, and usually they appear in lakes, of course, eutrophic lakes, but in lakes with stratification, summary stratification. And then we have another group that is the one called disperse through, that usually occupy the whole water column and is very common in shallow lakes. And these different groups, of course, belong to different species that have different traits and have different responses to the environment. Thank you very much. So let's go and see which are the species that appear in South America. And I just select a subset of lakes, and the numbers or the groups when the total cyanobacteria are quite high, just to see where they are, what is there. And this plot goes from the most abundant, the ones that have a higher biomass to the ones that have lower biomass, and this is the frequency of presence. So we see some names that are quite common for people that works with cyanobacteria, like Raphidiopsis, Plantotrix, Radiocystis. Microcystis is a little bit down there. And we can ask which of these ones appear reported in blooms. Who of them are considered like blooms and reported like blooms? And we have this second plot that comes from another work that we performed with groups of colleagues from the region. We did a survey looking for information about reports of blooms in South America and the Caribbean. Here we have the countries and the number of reports. The events of reports are indicated by the size of the circle here, and the color is related to the order, taxonomic order. So, of course, we have here at the beginning Microcystis, which is the most common bloom-forming cyanobacteria reported in the world. So that was not a surprise. But here we can see in all this group of the Nostocales that we have very different taxa there, and some of them were reported quite importantly. So in general, we can say that there is a quite common presence of Microcystis and some Nostocales. So I would like to have a few minutes to give examples of these two groups. First, we are going to talk a bit about Microcystis, and I bring the examples from Uruguay. Uruguay is in a subtropical region, and this is the distribution of Microcystis in the country. Microcystis is marked here in yellow. And we can, in this map, it also shows that Dolichospermon is quite common, and there is a co-occurrence of the two together that we are still investigating why. There are several experiments going on in my lab about this. It's quite interesting. And the other thing, if you look at this map, this is the map of the country, and if you are wondering if agriculture is an important activity in Uruguay, you are right. Agriculture is very important, and that's why we have eutrophication because of the fertilizers and cyanobacteria everywhere. These groups, and especially Microcystis, form accumulative blooms. The other important thing here, we are in a subtropical area, and the dominant landscape in terms of fresh waters are low-playing rivers and reservoirs, are turbid waters that don't have a very stable thermocline. So what happened with that in Microcystis? In this little video, we see a calm day in a very eutrophic reservoir. And you can see that the cyanobacteria, you see the water is like a mirror, there's no wind, and the cyanobacteria are there in the surface. So what we can have are situations like we show here, where, for example, this data from this year, from this summer, we have a very superficial thermocline with no wind, a very low wind, that is disrupt the day after with higher winds. So we have very superficial and very weak thermoclines. What are the consequences for Microcystis? here we have, for the same days, we saw the temperature. This is the profile for chlorophyll A and the percentage of cyanobacteria. And they accumulate the first day, and of course, they are more mixed in the second day. So these thermoclines that are very short in time, they have consequences for the aggregation and disaggregation of these colonies in the surface. And this might have consequences for the pigment acclimation. And so we wanted to explore a little bit more how they respond and how they adapt to this change in light from one day to the next. And that's why we start looking at the pigments. And here I show you the specific pigment absorption spectra normalized by chlorophyll A. And we did the analysis for 3 depths: the surface, near the thermocline point, and a 3 meters depth. And here we have the first day and the second day. And we have a big difference in general, as we can see, between the first day and the second day. And this is telling us about the self-shading and the aggregated colonies and photoacclimation in the first day in comparison with the second day. Recently we started working in collecting information and making an open data repository of PABs and CDOMs for complex and turbid waters for Uruguay. So for the ones that are interested, the data are there, and we wanted to, of course, enlarge the database. The second example I would like to talk about it in this second part of my talk is about Raphidiopsis raciborski. This is a fascinating species, and really we can have a whole session talking about it. It was studied as an invasive species. It's an expansion around the world. It forms dispersive blooms. It's extremely flexible. It has a great plasticity in incorporating nutrients, in changing pigments. And we've studied this species a lot in my lab. I would like to show you only two aspects. The first one is the distribution in the continent, and we work hard to complete what was the more recent distribution in the continent. And we are pretty sure that this subtropical area, except for some information that is not that clear, only genetic evidence, but the subtropical region is the limit of the distribution in the continent, in the region. And here we'll show you this map, where we compiled information about the production of toxins for Cylindrospermopsis. Some people probably know that Cylindrospermopsis has an extraordinary and very interesting phylogeography in the distribution of toxins that are really related with the expansion of the species. And for example, in Australia, this species produces Cylindrospermopsin and doesn't produce saxitoxins. However, in South America, almost all populations that are here marking different colors, but all of them summarize the same, they are toxic and produce saxitoxins or analogs. However, when we go to North America, that is much less information, the populations are not toxic. The other thing that's interesting is that there is a quite diversity or variety in the toxicity of this species, because, for example, here we compare in this study, this is a strain from Uruguay, this is a strain from Brazil, but it depends on the analogs that they have. The toxicity can change quite widely. Here we have two messages or two consequences. The first one is at the ecological level. The populations in South America that produce this toxin, they can have different effects in predators, if toxins are related with predators, or they can have consequences in interactions within the populations or with other organisms, with other species, considering the toxins as infochemicals. And it doesn't happen in the populations that are in North America, for example. So that is a completely different relationship with the communities. And the second one, of course, is about management. So clearly, for South America, this is a message. We need to monitor saxitoxins in the waters, because this is a very common species and produce saxitoxins. So closing to the end, we are going to talk a little bit about cyanobacteria monitoring. And I bring you another map to show what is the situation about monitoring cyanobacteria in the region. This diagram represents 3 parts: monitoring for water supplies, monitoring for recreational water, and regulations, if regulations are implemented for biomass and toxins. And the different shades indicates if they are or not in the country. And what we can see is that we have a very uneven reality, an uneven situation between countries. And it's this reality, which is quite different, it's even different between the countries that have everything implemented, which are only Uruguay and Brazil. So here, Uruguay and Brazil, for example, have different regulations about how to report the biomass of cyanobacteria. In Uruguay is with biovolume, but in Brazil they report cells and abundance. So the results are incomparable, and the consequences for management can be very, very different. So there is a lot of limitations here, and there is a lot of things that needs to be improved in the region. We need much more collaboration and much more interaction to come with a common framework that really could help the problem with cyanobacteria. The other thing I would like to add here, it's a personal note in the next slide, and it's the relevance of working with the community or the society. And I have the experience only from Uruguay, but I believe in other countries in the region is similar. We have a quite good I will say, communication or exchange, that is a better word, exchange with communities. Here is just a picture of an event that was organized from a community that invites scientists to talk about cyanobacterial blooms, all the problems and how they can help and solve. So here we find that usually there is a quite good interaction, but we are having problems to reach these people, which are the ones that can change, for example, regulations. And I think that is because the pressures they have are different from maybe taking care or reducing eutrophication. And if we see the challenges for South America about this point, we have very bad news. And we had good news for the blooms. Cyanobacteria will be happy there, but we have very bad news for our ecosystems. The first one is agriculture. The predictions for the region are more agriculture. Just looking at this map, this is the total fertilizer consumption between 1993 and 2023 in the world. And if you pay attention, South America is one of the regions that increased the most in the consumption of nutrients, fertilizers. On top of contamination, we have river fermentation and more reservoirs coming on top of rivers, and of course, finally, climate change that will make all this scenario much more difficult and much more complex. So this is what I wanted to share with you today, and I leave you with these take-home messages. Thank you very much for your attention. According with the clock, I have 2 minutes for questions, or 2 minutes and a few seconds. If someone wants to ask a question or a comment, is welcome. You need a microphone. No?
I'm not in. Okay. No, I wonder. I'm not a cyanobacteria specialist, but I wonder what is the reason behind this invasive species that produce toxins in South America and not in North America?
Yeah, it's a great question, and it's a very, very interesting question. It seems to be that it's related with the evolutionary trajectory of the species. So there are different routes where the species, there are different theories about where the species originated in geological times, and then how it spread in the world. And there are some different clades within the species that emigrate to different continents. And it's supposed to be that lost the capacity or acquire the capacity to produce one of the two toxins. And so it's a very interesting pattern. There are no non-toxics in North America, saxitoxins in South America, cylindrospermopsin in Australia. In Europe, there are no toxins, or I don't know if there is a recent report. So it's really, there is a geographical pattern of this species. And it's not only related with toxins, because when you start studying and comparing strains from different latitudes or different parts of the world, they have differences in growth rates, in nutrients uptakes. So it's really, I remember once Komarek visited Uruguay a couple of times, and he was super interested with this species, and he said his hypothesis was a species in speciation. It really was diversion, like in different ones. Even there are differences, genetically differences between strains and ecotypes.
Thank you very much for your wonderful presentation.
Thank you.
Thank you, Sylvia, for this wonderful presentation, and congratulations for your awards. Hi, everyone. My name is Romina Schiaffino from CONICET, Argentina. It's a great pleasure to introduce Hilary Close, Associate Professor in the Department of Ocean Sciences at the University of Miami Rosenstein School of Marine Atmospheric and Earth Science. She specializes in marine organic geochemistry and the ocean carbon cycle. Her research focuses on how microbial processes and particle dynamics regulate the production, transformation, and export of organic matter to the deep ocean. She's an expert in the development and use of compound-specific isotope analysis. Her work has advanced our understanding of the biological carbon pump and its role in long-term carbon storage and climate regulation. The plenary of her presentation is Hidden Beneath the Surface, Unveiling the Distinct Biogeochemistry of the Lower Eutrophic Zone. Please join me in welcoming Professor Hilary Close.
Okay, thank you so much for this opportunity to the organizers, for giving me the opportunity to fill in for my colleague Mak Saito. This will be a different topic. And I am switching to the ocean for most of this talk, but I do want to draw connections to lake systems that I think exist with some of what we're looking at. And I will actually start by just mentioning my pathway through science actually started here in Northern Ohio. I'm going to mention this for two reasons. One, it has a maritime border with Canada, which I never even thought about until I was getting a security clearance for something. And number two, I grew up on a family farm, and our drainage drains ultimately into Lake Erie. So I am really happy to talk with anybody working on lake systems and eutrophication. I'm still closely tied to our family farm there. And then, so those are the places that I have trained, and then these are the places that I have been able to conduct oceanographic research. And so I'm really accumulating the global ocean a little by little over time. And this is also a way of introducing one of the main ways that we can take a snapshot of the global ocean is by looking at satellite chlorophyll imagery. So I'm going to start there to introduce our main environments for this talk, which are the oligotrophic gyres. And I'm going to specifically be talking about the time periods of the year in which those gyres are thermally stratified. And the euphotic zone develops quite deep. So oftentimes we see that 0.1% surface irradiance reaching down to about 150 meters. And just in case anybody isn't thinking about this frequently, we do think of the euphotic zone as that part of the water column where photosynthesis is occurring at measurable rates. And so I won't be going beneath the euphotic zone very much today, but I'm happy to talk about that later. And so, of course, our ocean gyres, our subtropical gyres, are the main oligotrophic environments throughout the global ocean. And I have been making measurements in 3 of those gyres, one of them very recently, but I'm going to focus most of what I talk about today in the North Atlantic subtropical gyre off of Bermuda. And, but we are expanding these measurements, and we're starting to see a lot of coherence across these. Okay, and I'll try to mention as I go along why thinking about these environments and the patterns that I'm talking about are important. And one of the big things is that these oligotrophic environments are expanding as sea surface temperatures rise because of the increasing thermal stratification. So like I said, one very useful way to look at these environments is by satellites. We're looking here at annual average chlorophyll. And I want to start with the question is, what is it that satellites are actually seeing? And so we can look at a bit of a schematic of biogeochemical cycling in the ocean. And this is really just to point out that satellites see the surface. And so satellites now have data retrievals and various algorithms that have been developed that allow us to make a lot of estimates about the types of phytoplankton communities that are active and also some of the food web cycling occurring. Nonetheless, the observations are only in the upper few meters. And as I just said, in a lot of these environments, the euphotic zone can extend down to 150 meters. So clearly, this method is not going to directly capture all of the relevant primary production happening in the upper ocean. And so this was really the focus of the exports program, which was a program funded by NASA, and I was involved in it through partnership with the NSF. And this is one of the big diagrams from the exports program showing the complexity of both production and of recycling processes in the upper ocean, and then how that translates into what gets exported into the deep ocean and stored in the deep ocean or consumed, or potentially getting all the way to the sediments. So the idea behind exports was, in fact, to be able to better essentially ground truth what we're trying to predict from satellites by making very intensive in situ observations. Nonetheless, we are at a point that we still need to make in situ observations to really capture processes below that upper layer. So that's what I'm going to be focusing on today, is sort of the complexity that we can capture through those in situ observations. Okay, so in case you have not worked in oligotrophic systems like this, like I said, the euphotic zone tends to extend to around 150 meters, sometimes deeper. This is some data from the Bermuda Atlantic Time Series study site. And this is just showing some very basic properties of these types of water columns. Number one, our photosynthetically available radiation steeply drops off as the light is attenuated through the water column. And so we have a big difference in the amount of light that's being received in the upper and the lower part here. And incidentally, this dotted line is showing approximately where the mixed layer might be during stratified conditions. And then we have also very low nutrient concentrations. Here I'm showing nitrate plus nitrite in the upper part of the euphotic zone, and eventually those do increase. And really the two of those properties together lead into the distribution of phytoplankton biomass and the development of what's called a deep chlorophyll maximum. So we have a sweet spot where nutrients are more available and there's still sufficient light for photosynthesis, and then we have this peak in chlorophyll fluorescence. On the other hand, that is not always the peak of where productivity occurs. In fact, most of the productivity is occurring shallower. Nonetheless, photosynthesis is occurring at the DCM and below. And so we can generalize these habitats of the upper part of the euphotic zone as a high light, nutrient-limited habitat for photosynthesis, and the lower part as a light-limited and high nutrient habitat for photosynthesis. And then we get this response from the photosynthetic community of producing lower chlorophyll in this upper part, so we see low chlorophyll to carbon ratios, and more chlorophyll in this lower part of the euphotic zone. So those are some basic observations that are very widespread. And then, as I said, these data are from the BATS site, and Station Aloha in the Pacific looks very similar. And I just pulled some data from a really nice recent paper showing how the primary production rates are distributed over the same depths. And indeed, the primary production is actually higher rates in the upper part of the euphotic zone and lower rates under these light-limited depths. Nonetheless, if you integrate the total photosynthesis throughout the euphotic zone, this lower part is contributing on average somewhere around 20 to 25%. And what this recent analysis of the time series data from Station Aloha found is that, in fact, this deep photosynthesis in the lower part of the euphotic zone is actually more responsive to some climatological changes over the last several decades. So over these decades, we've seen an increase in primary productivity in this lower part of the euphotic zone and no similar detectable increase in the upper part. So this is one reason why it is certainly worth examining more closely what exactly is controlling the primary productivity in this lower part of the euphotic zone. So I have some other questions too, which largely relate to what gets exported out of the euphotic zone and how can we trace what has happened in the upper part of the ocean by what is sinking into the deeper ocean. Okay, so my work focuses around particulate organic matter, and the big question that I'm asking today is how does the composition of particulate organic matter reflect those biogeochemical differences between the upper and the lower parts of the euphotic zone, and also might affect the biogeochemical cycling in turn. And so when I talk about particulate organic matter, I'm talking about a big mixture of things. So when we collect particulate organic matter, or POM, we are capturing both the living and non-living components, unicellular components, and that involves both autotrophs and heterotrophs, photoautotrophs and chemoautotrophs. And with particles, there are a lot of physical dynamics to consider. So particles can sink through the water column, they can be produced as feces by zooplankton, and therefore get injected quickly at whatever depths the zooplankton are. And then we also have aggregation and disaggregation processes. So these are all things that we have to think about when we are trying to interpret the chemistry of what we measure in POM. And a couple other notes, what I'm going to be talking about today are some really specialized chemical analyses that reveal properties of POM and then biogeochemical processes of these parts of the euphotic zone. And also, we wouldn't really see what we're seeing if we did not do this at high depth resolution and also by collecting different sizes of particles so we can get at these dynamics. And so this involves some time-intensive sampling. So everything that I'm showing today was collected from shipboard sampling, and these are just some nice photos from a recent cruise I did just last month. Time-intensive sampling shipboard, labor-intensive analyses in the lab. And this is just something that I like to think about for the future. We're trying to get more global data through more autonomous platforms, and just thinking about in the future, what is the future of seagoing oceanography, and can we still support this type of discovery? Okay, so I'm going to focus what I show you today around 3 very simple observations, and then going into depth about what we think they're actually telling us. So basically, by making measurements over and over again in different oligotrophic gyres, some patterns started to pop out to me that I did not think had been fully described. And part of that was because we had a lot of latitude within our programs to actually do sufficient sampling to capture these high-resolution depth profiles through the euphotic zone. So in some sort of classical geochemistry, you might just hit standard depths, 50, 100, 150 meters. You might actually miss these sorts of features. So very basic parameters here, the C to N ratio of the particulate organic matter, the bulk natural abundance delta C13 value of the particulate organic matter, and we're seeing some big differences between the upper part of the euphotic zone, which I'm delineating here with this line, and the lower part here, and then this is the base of the euphotic zone here. So we've got some big differences that we'll dig into, and then this is a more specialized measurement that I will be talking about as well. Okay, so some of the big questions that come from these very simple observations of sort of big unrecognized geochemical differences between the upper and lower euphotic zone is, do these differences in the habitats for primary producers, the high light, low nutrient, low light, high nutrient, create different organic properties in what is produced? Then, if we have different types of organic matter, do we have different sort of responses from the heterotrophic community? And then what does that mean for the overall biogeochemical cycling? So, and then as I said, I have a long-standing interest in what gets exported, what's available for the mesopelagic food webs, and then also what gets recorded in the sediments and how can we interpret it. I won't get so much into that today, but I'll try to touch on it. So first, this is going to be a story time order, because this is the order that we actually sort of discovered these things. So I'll start with this pattern in the bulk delta C13 values of particulate organic matter. So the figure that I just showed was from the Bermuda site, and this is from some work that I had done previously at Station Aloha. Basically saw the same thing. And I will also just note here that this difference in the delta C13 value between the upper and the lower euphotic zone is huge. If you are, for instance, a geologist, you would think that this means some big shift in the carbon cycle if you were to see this in the sedimentary record. So to dig into this a little bit more, first we did a global analysis of whatever data were available out there. And so what we found was, we found this pattern over and over again in published data, and we found that this was global in the open ocean. So this was excluding ODZs, excluding places with a lot of riverine influence. So in open ocean locations, we saw this decrease in these delta C13 values between the upper and the lower euphotic zone, but what we also found was that there was no agreement about what caused this. And so the question going forward is this a essentially bottom-up signature coming from the photosynthetic community, or is this some aspect of degradation? And so just to add to our features of the upper and lower euphotic zone that I showed before, I've just slid in here some carbon parameters. So we see DIC concentrations, CO2 concentrations, and those increase between the upper and the lower euphotic zone. And then the delta C13 value of the CO2 that's used for the photosynthetic substrate also has a change over those depths. And so that is a consequence of the removal of CO2 by photosynthesis, removal of light CO2 driving this value heavy, and then respiration adding back in light carbon in the lower euphotic zone. So this is known. And so we suspected that what we were measuring in the organic matter was some consequence first of this pattern, and then some other features on top of that. So we really wanted to question whether there was a photosynthetic mechanism behind this. So what we did is we used compound-specific isotope analysis to isolate phytol, the side chain of chlorophyll, and that basically gives us an integrated picture of the whole photosynthetic community. If you would like to read more about compound-specific isotope analysis, I wrote a review about it several years ago. Has lots of good stuff. So this is the work of my former PhD student, Lillian Henderson, and the bulk pattern that I showed you is just here. It looks a lot smaller because what we found in phytol, which we cleaved from chlorophyll, was a much bigger decrease in those delta C13 values. So to boil this down rather quickly, what we can do is we can look at the difference between those phytol values and the CO2 values, which that was formed from by the photosynthetic organisms, and we can calculate the fractionation during photosynthesis. And so this is something that people who work in the geologic record think a lot about because it can be indicative of the conditions that the photosynthetic organisms were living in. So here, by demonstrating this in a photosynthetic compound, we showed that the origin of this bulk pattern is in fact photosynthetic. And then we put this in the context of existing work, so thinking about the dynamics of CO2 uptake by phytoplankton and their growth rates. And we could partially explain what we saw. So we see that it fits nicely. These are our data in the bold symbols. They fit nicely into the same patterns of data found previously. But then if we account for also the surface area to volume of our phytoplankton community, we now see that there is something else driving what we're seeing. And what I will point out is that our study was really the first to look at this over depth. So most of the rest of this had been done in culture or looking at these variables over latitude instead of over depth. So something different is happening when you look at this over depth. And so another broad scale thing I want to talk about today is the power of collaboration. So I am trying to feature nice photos of my collaborators, one of whom is right in front of me. So by having collaborators who are experts in phytoplankton community characterization, we were able to look at our data more in terms of the reality of what you see between the upper and lower euphotic zone. And so what we found, what we pulled out of that, is that there are factors that are not considered in a lot of these photosynthetic models, things like what phytoplankton are actually living at these different depths and under these different CO2 and nutrient conditions. And so the two things that we really pulled out that are not currently considered in these models is the community composition, and we found that small eukaryotes might have an impact on this, and then light itself. So usually in culture, the duration of light might be varied, but not necessarily the intensity. So we think the light intensity might have a factor here. Okay, so this, we think, is a potential tracer that we could use to examine whether material being exported out of the euphotic zone is coming from the upper or lower portion. So again, we don't really know the importance of that primary production in the lower euphotic zone to export unless we have a way of tracing the material, tracing the origin of that material. So we are still developing the possibility that these low delta C13 values could help us do that. Okay. So again, story time. We also realized while we were making these measurements, because we were doing all these estimates of the phytoplankton biomass as well as their delta C13 values, that we could do a mass balance calculation and also estimate the delta C13 value of everything that's not photosynthetic. So like I said, POM is a big mixture, and so we were able to calculate the delta C13 value of the other stuff, which would be heterotrophs and detritus. And so to bring this down to our conceptual figure from this paper, from our phytol measurements, we have the delta C13 value of the phytoplankton community. We saw that that decreased between the high light upper euphotic zone and the low light. And then here's our mass balance calculation of the other stuff. And basically what we find is there's a lot of similarity in the upper euphotic zone and there's a divergence in the lower euphotic zone. And so what we think this is telling us is that in the upper euphotic zone, heterotrophs are living in a close association with the primary production, which fits with a lot of other theory that we think about in the upper euphotic zone. But as we get into the lower euphotic zone, we have accumulated detritus sinking from above, and so the heterotrophs living here can rely both on the in situ production and this sinking material. And so we get a divergence in the geochemical signatures of the photosynthetic and non-photosynthetic material at these depths. So this also then led us to question, well, what else might we discover about the heterotrophic metabolisms of the lower euphotic zone? Are there other things to look for that might shed light on what they're doing there? And so this leads us to this more complicated measurement, which is a compound-specific measurement for which we estimate trophic position. And I'll try to explain what trophic position means in particles. We measured the delta N15 values of individual amino acids, compound-specific isotope analysis of amino acids. And the basic theory here comes from animal food webs. So in animal food webs, the internal digestion of amino acids results in the increased delta N15 values of some amino acids of the consumer compared to its food, and no increase in other amino acids. So because that is such a predictable difference in that increase and no change across specific amino acids, this can be used to calculate trophic position. Again, this is developed in animal food webs. And so you can do something like this. You can relate the actual trophic position of an animal to the delta N15 measurements of individual amino acids, and therefore you can just use those measurements to estimate a trophic position. So how does this relate to microbes and POM? You have to think about what actually causes that isotopic enrichment. And so what causes that enrichment is there has to be some release of ammonium during a partial catabolism of amino acids. So by isotopic mass balance, the only way you can get isotopic enrichment in something like glutamic acid is if there's a net release of ammonium waste. So it is the light ammonium that is excreted, leaving the residual pool isotopically heavier. Okay. So if we think about it that way, that means that when we see an increase in trophic position, that means that somebody has done some catabolism of amino acids. When we're thinking about bacteria, however, heterotrophic bacteria don't have to do that. They don't have to act like an animal. And so this has been shown in culture, and it has also been shown not isotopically in culture, that if heterotrophic bacteria are growing on a nitrogen-poor substrate, they might not produce any nitrogen waste at all. Instead, they are essentially recycling the amino acids that have been provided to them, for instance, by the photosynthetic community. So what we found in the same exact samples where we measured that phytol is we were able to make that measurement, calculate a trophic position, and because we had a mass balance estimate of the heterotrophic or the non-phytoplankton component, we were able to estimate the trophic position of the material provided by the non-phytoplankton component. And what we saw is that increased over depth. And so what this boils down to is that as depth increases between the upper and lower euphotic zone, amino acids are being catabolized more in the lower euphotic zone, meaning they're being used for energy, whereas in the upper euphotic zone, they appear to be recycled efficiently and not used for energy. And so what that means is the implication is that there would be ammonium waste generated in the lower euphotic zone and not as much on a net basis in the upper euphotic zone. Okay, and we also found this in our one of our exports locations. So that was work by my former PhD student, Paul Voital. You can read that there. And so adding on to this picture, we now have this estimate of the trophic position of the material. And again, we're seeing that divergence in the lower euphotic zone. So again, a tight coupling between the photosynthetic and heterotrophic communities in the upper euphotic zone and divergence in the lower euphotic zone due to greater reliance on catabolism of the more abundant detritus at that depth. Okay, so then we thought about, now that we're thinking about ammonium release and that sort of thing, how does this relate to the broader biogeochemistry of the upper and lower euphotic zone? And then we get to this very simple observation of, but very robust observation of a decreasing C to N ratio between the upper and the lower euphotic zone. So this is continued work by my former PhD student Lillian, and what we found when we aggregated our elemental data from a bunch of cruises is we had these very beautiful profiles showing a very consistent pattern of high C to N ratios in the upper part of the euphotic zone, there's the dividing line there, and lower in the lower euphotic zone. And so our hypothesis was that this had to do with the biochemical composition of the organic matter, and with help from another collaborator in Craig Carlson's lab, we analyzed the carbohydrates in those very same samples, as well as the amino acid abundance. And what we found is that that varying C to N ratio, in fact, was largely driven by how much carbohydrate and how much amino acid was in these samples. And so what that means is we have a high production of carbohydrates in the upper euphotic zone and a higher proportional contribution of amino acids in the lower euphotic zone. So putting that together with what we were just talking about, that means that in the upper euphotic zone, there seems to be exactly that situation where heterotrophic bacteria would be metabolizing nitrogen-poor organic matter and not necessarily producing a Redfield balance of nitrogen waste. And so putting this side by side, this was our trophic position of our non-phytoplankton parts of the POM. Our hypothesis is in the upper part of the euphotic zone that amino acids are going to be recycled for cell growth instead of catabolized for energy. And then in the lower part, the carbon is going to be oxidized for energy. The nitrogen is going to be expelled as waste. And this is also going to provide fuel for nitrification, which takes off in the dark right around the base of the euphotic zone. And that's going to produce additional oxygen demand during that oxidation as well. And so I just showed you this in a cross plot, but here are the depth profiles of the carbohydrate abundance and the amino acid abundance. And so we do see that higher carbohydrate proportion in the upper euphotic zone. And so we hypothesize that these are being used more for energy, along with energy supplementation through photoheterotrophy. So this is kind of the overall hypothesis that has come out of these detailed geochemical observations, that there's community nitrogen limitation and catabolism of abundant glucose in the upper euphotic zone, and in the lower euphotic zone, more of that amino acid is available for energy usage, and that produces that ammonium waste. So this is our hypothesis, and now we get to test it. And so with further work from the Bioscope project, which you can read more about there, we've been digging into this. So this is work by Jackie Comstock, looking at the heterotrophic prokaryotic communities. And I'm highlighting here the depths that I was just talking about. So the upper euphotic zone communities are really, really distinct from the lower euphotic zone communities. So we're not necessarily looking at their metabolism, but we can see there's definitely different communities there. To look into those metabolisms, we're starting to look at the metagenomic capabilities at the different depths. We can look at their proteomes with aforementioned Mak Saito. We're looking to do some cell sorting techniques and also to make sure this makes sense with what we're seeing in the DOM pool as well. And then there's some broader implications that we're interested in looking to in terms of the stoichiometry of remineralization. As well as what that means for what nutrients are available for photosynthesis, and then what models are going to be affected. So models, for instance, that assume the Redfield ratio are going to be highly affected by these findings. And the continuing work on this project in my lab is by Dalen Jing, so look out for more of that. To put this all together, we layered this carbohydrate data on top of the isotopic data that I described before, so more glucose up in the upper euphotic zone and low carbohydrate in the lower euphotic zone. And I'm just going to put those up really quickly, our hypotheses. And just also note that the implication of our hypothesis here of greater ammonium release during amino acid catabolism in the lower euphotic zone would also mean fuel for ammonium oxidizing archaea. So that leads us now to what is exported. So what's exported to the deep ocean to fuel the food webs there? What's exported to the sedimentary record? The pictures I put up here are to remind me to talk about mechanisms of export. So this POM can get exported if it aggregates into large particles that can settle gravitationally, and also if it is consumed in the metazoan food web and packaged into feces. So as we think about what is exported, we also need to think about where zooplankton are feeding in the water column and where aggregation is occurring. And so the question that you can think about, even if you work in other types of stratified systems, is if you have geochemical differences, say between the upper and lower euphotic zone or a chemocline or something like that, what are the export processes that are layered on top of those differences? And which of these signals is actually going to get into your deep ocean food web and into the sedimentary record? So some of the ways that we're starting to look at that, one of my former graduate students, Shannon Doherty, used amino acid compound-specific isotope analysis to essentially develop a signature of fecal pellets that's distinct from phytoplankton aggregates or microbial biomass, and therefore might give us a tracer of where fecal pellets are being produced in the water column. And so this is just an example from Jackie's paper that I brought up before, where we found in these large particles in red, my depth axis got cut off, sorry, this is the base of the euphotic zone, so this is in the lower euphotic zone. What we found is a high trophic position and these low delta N15 values of threonine. Both of those indicate that fecal pellets are showing up in the lower euphotic zone specifically. So this is giving us the idea the lower euphotic zone actually might be a hotspot for fecal packaging and therefore might actually efficiently package all of those geochemical signals from the lower part of the euphotic zone. We also, through these methods, and because we look at size fractionated particles, we get signatures that might relate to particle disaggregation. So what we see here, just below the euphotic zone, is those same fecal pellet signatures showing up in our smaller particles. So we're also starting to dig into whether right below the euphotic zone is a hotspot for particle disaggregation. So we kind of need to think about all of these when we think about what is making it to which depths of the water column. And there's a bunch of results coming from the exports program around this type of categorization of particles. One is published by my former student, Paul, and there's a couple in the works from my former student, Liz. So be on the lookout for those. And then also, two of my new projects are also really digging into this question. Now that we are developing these really different signatures from different parts of the upper water column, how can we track those into what is exported so that we can understand better how production in the lower euphotic zone actually translates to carbon sequestration or to the sedimentary record? And so these are two projects with the Schmidt Sciences Ocean Biogeochemistry Virtual Institute. If you haven't heard about it, it's 5 really big collaborative projects. So I'm just showing a couple of my collaborators here. So one of these projects, we are basically expanding our characterization of feces to all kinds of animals, all kinds of depths in the ocean to get a better signature of where fecal packaging is occurring. And then the Subsea project, which is headed by Matt Church at University of Montana. This is explicitly focused on the lower euphotic zone and its importance in biogeochemistry. So we have a bunch of collaborators, a lot of people in Hawaii associated with the Hawaii Ocean Time Series, and we are really, really digging into the specific nutrient budget of the lower euphotic zone. So be on the lookout for work from those as well. And I will just acknowledge a bunch of people from my lab over the years who have contributed to this work. We do have another new project that is looking into the fundamental aspects of isotopic fractionation during these bacterial amino acid metabolisms. And I am currently advertising for a postdoc. And then thanking my funding, largely from the Bioscope program from what I've shown you today. So I think I have a few minutes for questions if anybody has any. Thank you. And also come talk to me about Lake Erie fertilizers at any time. Yeah.
Hi, thank you, an amazing presentation. And in the subtropical gyres, the upper part is dominated by picocyanobacteria, Prochlorococcus and Synechococcus. And in the DCM and below, there's an increase in eukaryotic algae. And cyanobacteria accumulate mostly polysaccharides as a reserve energy, while eukaryotes can shift to accumulate liquid droplets, for example. Is there any effect in the isotopic fractionations due to the upper dominated cyanobacteria community and the lower eukaryotic dominated community?
Yeah, so that's a great question. In terms of the cellular mass balance, basically all the CO2 that comes in, there has to be an isotopic mass balance to all of the biochemical products of that organism. So the way that we think about it right now is that the net biomass is still going to resemble, it's going to reflect the isotopic fractionation during photosynthesis. The thing that I think might really affect that, which nobody is looking at, is how much of that primary production actually goes into the dissolve phase. Because if there is dissolved production that we are not capturing, then we don't have an actual isotopic mass balance. So that is one of the things that we mentioned in our paper, that dissolved production is a big variable. We didn't see at our study site a huge difference in the lipid composition. But another thing I think that is a huge lever on this is the expression of carbon concentrating mechanisms. That's one of our, basically another big lever on that isotopic mass balance. So I think this is largely a question for people who are trying to make really great models of carbon isotopic fractionation, and that is largely why they don't exist in a perfect way just yet. There's a lot of these variables. But yeah, I think especially looking at that really high carbohydrate content in the upper euphotic zone, we really want to dig into like what form those carbohydrates are in actually. I think there was a question.
Thank you very much for such inspiring talk. It was for me nice to see that the amino acids that are recycled in the upper photic zone can be attributed to the photoheterotrophic organisms. And the question is, did you manage to decipher what type of photoheterotrophy is the one driving this effect, since the two main types might have different nitrogen requirements?
Yeah, so the quick answer is no. That's basically next steps is looking into how photoheterotrophy could play a role. I think a really interesting thing that we can do is try to put together an energy balance, basically, for, if we think about the light availability at different depths, can we include that as part of the energy balance of heterotrophy? But I guess another thing to mention is we're starting to work on incorporating biomarkers for photoheterotrophy, like proteorhodopsin, as yet another marker that we can use to attach to these different processes. But yeah, to come in the future, I guess. Else? If not, thank you so much.
Okay, with that, I want to thank Drs. Bonilla and Close once again. I want to remind all of you about the rainbow reception, and thank you all for being here. Good night.
We are